Stream metabolism is a measure of ecosystem function based on primary production and respiration, basically the activity of the base of the food web. It is an experimental concept that has existed for decades but is becoming more widespread as technology is more affordable and durable. Relatively long-term data can now be used in monitoring to describe behavior of stream ecosystems over time. Modeling stream metabolism allows us to describe the sum of the metabolisms of all living organisms in a stream. Stream ecosystem metabolism is the ratio of primary production, oxygen produced by photosynthesis, to respiration, the oxygen used by organisms during metabolism. In small streams, this is largely driven by biofilms that include algae and bacteria as well as aquatic plants. These primary producers fuel the higher levels of the stream food web. Just like terrestrial plants, aquatic plants like warm temperatures and lots of light and nutrients. So primary production in a stream is highest during the summer when there are plenty of resources to fuel the stream's producers. On the other hand, metabolism is lowest in the winter when primary producers are dormant and the stream is cold.
While these direct causes of stream metabolism seem straightforward, there are many factors that are interconnected. The regional geology drives the land use in the area, which affects the direct drivers of the stream metabolism, like hydrology, sediment, light, and nutrients. For example, some areas in the Midwest are well suited for agriculture, which has major impacts on stream function. Here we see two streams in the Green Bay, Wisconsin area that are impacted by some nearby fields, Turtle Creek and Sugar Creek. The activity tends to increase the available nutrients and sediment in the stream through fertilizer runoff and erosion. When the forested areas near the stream are cleared, light and temperature also tends to increase. Hydrology also impacts metabolism through variation in stream flow that can scour stream beds. But on the other hand, forested streams like Kirshner Creek receive less direct sunlight and nutrient inputs than these agricultural streams. The stream metabolism is likely lower in these streams, which depend on dissolved organic material from the streamside riparian zone.
Measurements of stream metabolism rely on a mass balance of dissolved oxygen. There are 4 factors that drive the concentration of oxygen in a stream. Primary production creates oxygen through photosynthesis, while respiration uses oxygen as organisms maintain and grow. Groundwater inputs can contribute water with low dissolved oxygen, while atmospheric oxygen can be incorporated into the stream as water runs over riffles and is aerated. 2 major parameters of the stream must be estimated to model stream metabolism over a certain reach: the reach travel time and gas reaeration coefficient. The reach travel time, paired with logger data, is used to describe how production and respiration affect oxygen levels as a specific parcel of water moves through the reach. The reaeration coefficient corrects for the atmospheric oxygen that is dissolved into a stream. To calculate gas reaeration over the reach, propane is used as an analog for oxygen. Propane bubbles out from the stream at a similar rate that oxygen is dissolved from the air. We used a grill tank with a large air stone and tarp at the upstream end of the reach. Water samples were collected by syringe and evacuated vials and run on a gas chromatograph in lab. The reaeration coefficient K is described by the decreasing propane concentrations over the reach. To measure reach travel time, salt is injected at a steady rate at the same time as propane. This paired tracer injection allows for calculations of average stream depth and discharge, which are required by some models. Measure the stream wetted width at 2-meter intervals along the reach if you want to calculate average depth. A peristaltic pump was used with a solution of uniodized salt in stream water to increase the conductivity of the stream by 10 to 15 microsiemens per centimeter. Conductivity meters at the upstream and downstream stations recorded the salt plateau as it passed through. The target reach travel time for the two-station method shown here is 20 to 30 minutes. In this case, water took 23 minutes to pass through a 50-meter reach. After the data is collected to calculate reach travel time and the reaeration coefficient, 2 logger stations are deployed. At each station, a dissolved oxygen logger is suspended at half of the total water depth, with a light logger at the top calibrated to convert absolute light intensity to PAR, or photosynthetically active radiation. A depth logger at one of the stations recorded any changes in water level. Stations were set to record data points every 5 minutes and left in the field for a week during late October. Here you can see the output from this experiment. The days that were sunnier and warmer showed higher rates of primary productivity. Stream metabolism was higher, as we'd expect. Modeling stream metabolism is becoming an important tool for monitoring restorations. Stream biofilms and macrophytes can respond to change on very short time frames, making metabolism a useful metric of restoration success. Mahone Creek, a tributary of Green Bay, is a potential target for stream restoration activities to increase fish passage and decrease impacts of stormwater runoff. Tracking stream metabolism could complement other monitoring actions to describe stream function more completely.